Today we will discuss about the most important topic of chemistry. It is stereochemistry. So what is stereochemistry? Stereochemistry means two words, stereos and chemistry. Stereos comes from the Greek word solid, and chemistry means the spatial arrangement in 3D. Actually, stereochemistry means involves the spatial arrangement of atoms in 3D. First stereochemist was Louis Pasteur. The development of stereochemistry in its present form is not very remote. In the last 50 years, chemists of all branches have contributed in a big way to make the subject fascinating, and at present, no chemist can afford without a reasonably detailed knowledge of the topic. The development of computer as an investigations of structure of complicated organic and inorganic molecules have raised the subject to a new height and newer conception and enriching it every year. Stereochemistry is now the subject of study in every branch of chemistry, but because of the structural variations of organic molecules, the subject has penetrated more deeply in organic chemistry. That is why stereochemical investigations are mostly done in organic molecules. We cannot see an atom of carbon, but when a large number of carbon atoms join in specific geometrical order, we get the most precious jewel called diamond. Thus, when we can say that people are charmed by diamonds, they are in fact attract not its constituent atoms but by their unique arrangement in space, that is stereo arrangements. A particular type of protein molecules within our body plays specific biological role because of their unique three-dimensional framework. If these shapes are lost or distorted, their biological activity is found to be absent and they are no longer useful within our body. In fact, the very existence of life on Earth deeply depends on the geometric shapes of organic molecules, which are basically made of few elements like carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus. Therefore, we can conclude that atoms can produce some unique properties only when they are arranged in specific geometric shapes. But it is very unfortunate, or unfortunately, we cannot see molecular structures in three dimensions. But the three-dimensional structure was accepted with increasing confidence during the latter part of the 19th century as classical hypothesis. Pasteur, Van 't Hoff, and the others successful explanation currently predicting many cases of isomerism in organic molecules on the basis of this hypothesis. Of the introduction part. So, what is the terms related to stereochemistry that you must know to submit or to prepare for a entrance exam? Now, some terms related to stereochemistry are dynamic stereochemistry. Dynamic stereochemistry involves the effect of stereochemistry on the rate of the chemical reaction. Next, plane polarized. Plane polarized, it is nothing but there is a machine called polarizer machine or polarizing machine. What it is done? It converts the normal light or the light that comes from the sun, it converts it to a polarized light. We know that in normal light or sunlight involves many types of radiations or many ultraviolet. Many radiation means it involves many radiations of different side, but it converts to a one particular side. This is the mechanism of polarized light or plane polarized machine. Next, optical activity. Optical activity is the interaction of chiral molecules with polarized light. When plane polarized light passes through the chiral molecules, it converts or it's able to converts it to a polarized light. But if the and we see the molecule is optically active, and but if we pass a plane polarized light through a achiral molecule or the molecule which is not chiral, then we the plane polarized light cannot pass through it. Next, optically active molecule. Optically active molecule are non-superimposable to each other. What is non-superimposable? Non-superimposable means not superimposable. That this means that human hand is the best example of it. Two human hands, left hand and right hand, not be superimposed to each other, and if this happens, then we can say that the two molecules are optically active. Next is asymmetric atom. Asymmetric atom means lack of symmetry. We will discuss about asymmetric atom or asymmetric carbon center or stereogenic center later. But you must consider it now that asymmetric atom means which there is no symmetry. Asymmetry means no symmetry. So if a atom consists four different atom means here we consider carbon atom, because in stereochemistry we will discuss about all mainly carbon atom. So if carbon atom consists four different atoms, then we can say that carbon atom is asymmetric atom. Next, we sometimes then not say perfectly that lack of symmetry means asymmetric atom. The best example is mesotartaric acid. We will discuss about the mesotartaric acid later. Next, what is the significance of the stereochemistry? Significance of stereochemistry is very important in the human life. One of the most famous significance of stereochemistry was thalidomide diester. Thalidomide is a drug. Its R isomer was prescribed for treating morning sickness in pregnant women, but the S isomer had tetragonic effect, causing a serious genetic damage to damage the embryonic growth and development. There is the structure, you can see it. See that are the two structures are about same but not. Here is the this part is above the plane, here this part is below the plane, and this different configuration changes the contradictory effect. Discussed till now is not important from exam point of view. What I will be discussed from now is important from exam point of view. Now, what is isomers? Isomers are same molecular formula but different physical and chemical activity. If we consider these two different and physical and chemical activity molecule, but they have same molecular formula C2H6. So two different molecule. One is ethyl alcohol and another is methoxy ether. These two different molecules have same molecular formula C2H6O, but they have different physical activity. They have different molecule. One is alcohol and another is ether, and this is called isomers. If Now, isomers are mainly two types. One is structural or constitutional, another is stereoisomers. These are structural or constitutional isomers. Structural and constitutional isomers are different connectivity. Their connectivity is different. But stereoisomers or optical isomers are connectivity same, or they are same connectivity. These two different molecules, orthoxylene and metaxylene, are same molecular formula but their connectivity are different. One is connected with ortho and another is connected with meta. But here in stereoisomers, their connectivity is in both cases are same in 1,3 position. But here 2 is above and here 1 is above, 1 is below. Now what is above, what is below, I will tell you. In stereochemistry, if the bond, if the connection is taken through a bold line or black line, then we say this is the above the plane, and this is above the plane. Means the atom is above, and the if dashed are signifies the below. This is below. This is above the plane, this is below the plane. Next, stereoisomers. Stereoisomers are two types: configurational, conformational. Configurational requires high energy barrier and here bond breaking is required, but conformational isomers are in low energy barrier. There is single bond rotation. Configurational isomers are generally two types: enantiomer and diastereomer. What is enantiomer? What is diastereomer? The definition, their activity, their all their questions related from enantiomer, diastereomer we will discuss about later in the next video. And configurationals are single bond rotation, were easily transferred, were easily converted from one conformational isomers to another easily. Their energy is required very low. This example, eclipse staggered. What is eclipse, what is staggered, what is cis, what is trans, we will discuss about the next video. So thank you.